will there be a community of biological hackers? And implicit in this is a community that helps make sure or promotes the overwhelming constructive hacking of biology. And to make this more specific, I don't know that I can do this. And I know like 3 people in the room here. Is the CCC a good organization for brokering a conversation about what the standards of community or practice would be around biohacking going forward? Right? You can see that we're not very good at it right now, but we're getting better very, very quickly. And so I think the time to have those conversations started is today. Thanks very much, and happy to take questions for the time that's remaining.
You were talking earlier about shifting the parts of the 29K thing. You did this with a certain intention in mind, but did you try it randomly? Because the lumberjack example that you gave, they were working. They were not working not as good as before, but they were working. Did you try it randomly and see if they were still working?
Yeah. So when we redesigned the bacteriophage genome, we did it in a very forward engineering, naive, deterministic sense. What if you just did it randomly, or what if you just evolved it, right? You could set up an artificial evolution experiment in the lab to evolve the virus to do different things. We have done that, and when you do that, you can get evolved versions of the virus that are better than the natural isolate, but we don't know how they work, and we wouldn't know how to forward engineer that. So there's a lot. I mean, seriously, if you like reverse engineering, I think the best set of reverse engineering problems in the world today is in genetics, because you have these reproducing machines that have been evolved for billions of years. Nobody understands them, and if you can make any contribution to figuring them out, because you're a great reverse engineer, you'll be celebrated all the way to Stockholm, if that's what you care about, right?
Yeah, I very much liked your talk. My experience is that I was a reverse engineer using Zilog microprocessors in the 1980s, shell scripts and C programs in the 1990s, and some Perl scripts into the 2000s. But I've quit with that. I'm no longer programming programs. I've come to resort to natural language and to socializing and communicating, and find that more effective. And what I've been studying is not so much the DNA. I take that as given, but I'm studying the human psyche and the emotional algorithms. And what I try to figure out what makes sense, what are emotions good for, and what is informational medicine. And I think I see a need that we must disassemble the algorithms of the molecular algorithms that happen in the neuron and in the limbic system. That control, that steer organisms the size of a human being. And I don't think it's necessary to fully grasp the DNA workings for that. Yeah, that's just what I wanted to share.
So I think those are some interesting comments. Obviously, understanding the human mind and how it works is one of the great scientific questions of our day. And it's interesting to imagine that we can make progress on that. There is... I don't know very much about that, but I do know of work coming out of the Rockefeller that suggests that many aspects of human memory or behavior are under direct molecular control, and so that it might not be possible to do a full abstract decoupling of organismal behavior from the molecular level, but that'd be interesting to see. Yeah.
I was just wondering, so I think it's a pretty interesting prediction to say there's going to be sort of a hobbyist community building stuff, and I was just wondering, to what extent can you already say it's even possible to regulate what those people will be able to build? Can you predict behavior of such a system based on the components you use. To which extent is that predictable?
I don't even know how to begin predicting that. I think and I don't know if it'll happen or not. I mean, my starting thoughts sort of, if I look at how I understand the personal computer to get going, that was pretty exciting. Here we've got a technology which is living, reproducing machines. It's a nanotechnology that works. So you could go hack silicon and software, and that's awesome. People should be doing this. But I think this technology is more powerful and more exciting and harder to predict, right? So I don't know exactly how that's going to play out.
So just to give a basic example, let's say you're allowed to build organisms that live but that don't reproduce.
Yeah.
Is that something that you can already do? Is that something that you can predict based on?
Yeah, so if you go back,
sequence?
that's a good question, right? So if you go back 30 years to the conversations where early genetic engineering and recombinant DNA technology got wrapped into biosafety, there were a lot of discussions about how to cripple engineered organisms, basically, so that they couldn't replicate or could only survive in certain environments. This has been very useful from a safety perspective, but it's been extraordinarily controversial from an economic perspective, because when you now go and look at things like agriculture biotechnology, those same features, which keep things from hopefully propagating out of control, also force a subscription economics model on farmers, because you have to keep going back and buying more, right? You can't just grow more. So they're interesting trade-offs there. I guess the meta lesson, the simple one, would be you have to pay for control basically somehow, right? So it depends probably how much energy you want to invest in making the systems controllable. That'll frame how predictable things are or not.
But generally, as soon as you have a sequencer, you can build everything, and it's probably not feasible to regulate the production of those sequences in a way that people won't be able to build arbitrary stuff.
The synthesizers, yeah. We just put out a 200-page study on options for governance around DNA synthesis technology. We looked at everything from controlling the regulation of growth of sugarcane to the distribution of the phosphoramidite bottles, those chemicals, to tracking who's placing orders and stuff like that. Not to recommend one or the other, but just to sort of say, if you look at the whole chain of technology, reagents, and knowledge, are there points of intervention that make sense? My personal conclusion from that experience is there aren't a lot, and it's much more important to have conversations like this so that we could frame the human community around the technology, because that's probably going to have a greater impact than any bit of screening software that's trying to infer the intent of somebody building a piece of DNA, right? So, I mean, the question to the CCC is there an opportunity to lead a conversation around how to promote a constructive hacker culture in biology and biotechnology is a real one. And if I can, get a response to that offline, I'd be really grateful.
Hello. Yeah, very nice talk. I don't know much about DNA stuff before, and I was very surprised about this talk. But the thing I was surprised the most was this machine. You call it the genetic synthesizer.
Yeah.
And can I make a few words to this thing, because it sounds to me like it works like a printer, or it's really strange, or it's not imaginable for me how it works. How much does it cost, and how long does it take to make one gene?
Yeah, those are good questions. So the best DNA synthesizers in the world right now are operated by Agilent, and the technology they use is an inkjet printing technology, right, to print the chemicals on different spots. And so basically the way it works is you're growing a polymer, you're growing a chain. Add an A, then a G, then a C, then a T, then a T, then a T, then an A, then a C, then a C. And the chemistry to add each of those letters is a 4-step chemistry. So you just sort of cycle through this 4-step process every time you want to add a letter. So if you want to build something that's 20 bases long, that's 80 chemical reactions. The chemistry was really well worked out in 1982, so it's 25 years old. And even though the chemistry is really old and really well worked out, you can see that it's hard to grow a polymer, if the chemistry was, I was going to go onto eBay. If you just type DNA synthesizer into eBay, you'll get the latest pricing. You could build oligonucleotides off a synthesizer that'll cost you $4,000. An oligonucleotide might be up to 100 units long, okay? And the reason you can't build more than that is every time you add a letter, the efficiency of adding a letter is 99%. It's really efficient chemistry. But then when you get out to 100, the overall efficiency of your process is 99% to the 100th power, which is like 30% yield. And so you just can't build longer than that. And then you have to figure out ways to stitch together bigger pieces from that. So it takes about 4 hours to build an oligonucleotide. The best oligonucleotide synthesizers can make 50,000 oligos up to 200 units long with an error rate in 1 in 250. So it's about $1 a base pair basically to assemble a gene or a genome today. And it'll take you, if you were to order a gene online, it would take you about a month to get it back with a 50% reliability rate.
Okay, last thing, is there just a USB version or can I plug FireWire too? No, sorry.
So I would like to have a machine called the StinkJet, which is a desktop DNA synthesizer. It would be, I guess it'd be FireWire. And what it would do is it would print 12 million base pairs of DNA a minute, double-stranded. And that would let me have a yeast genome every minute coming off my DNA printer. The economics of the marketplace right now are not supporting distribution of the synthesizers. It's going towards centralization of factories. And it might be later that you get a sort of fan out of desktop synthesizers a decade from now. Don't know. Yeah. So the question is, how hard would it be to take an inkjet printer and make a DNA synthesizer from it? I don't know. I mean, it would be hard for me, but I know that I could build a DNA synthesizer from off-the-shelf components that wouldn't be based on inkjet for under $10,000. That's probably not very good, though, at that price. So the inkjet stuff would be a little bit trickier, I think. But I bet you could figure it out if you wanted to. Yeah, so the question is, will schematics for DNA synthesizers be made publicly available? Let me respond to that specifically, but also from a bigger perspective as well. I think that DNA synthesis is really important technology. Whether you like the Star Trek comparisons or not, you can make genetic material from scratch. So it just matters, right? Who controls that technology, who owns it, who has access to it, right? Because those are the people who are going to be building genetic programs. There's very little public investment in DNA synthesis technology. Almost all of it is private. So Agilent has the inkjet technology. Other companies have other DNA synthesis technology, and I'm involved in one company because it was the only path forward for me to get the stuff, right? It's an important issue, and I guess you could reverse engineer a machine off of eBay, but I've not seen schematics. And as I mentioned in my talk, the documentation for the food replicator from Star Trek is better than the documentation for DNA synthesis. And I don't think that's good. I really don't. I think it needs to be a publicly accessible technology.
I would want to say that I think that both communities, computer science and biology, are somewhat mixed right now because there is some bioinformatics group evolving right now. Perhaps at our university it's quite a big community there. So I would ask you if that would be a good combination for both communities. And another follow-up would be, because most programming languages are based on a context-free grammar. I would like you, if there would be some, if there had been some research into that, whether